Good morning, everybody. Well, we had a really interesting time yesterday talking about sequence variants. I hope that you enjoyed that topic and thought it was really interesting. But you probably have some questions about where all these sequences we compared to, where this reference annotation comes from. And so today's lecture is about how we start with just the organism in question and make our way to an annotated genome for it. So the questions of assembly and annotation will be central to today's lecture. We will be starting with assembly, as I said. We're going to talk about a way that you can design your sequencing experiment so that you're able to more readily assemble it, the paired-end sequencing strategy. After that, we'll talk about the problems of junk DNA or repetitive DNA in the assembly process. From there, we'll talk about k-mers. This is a topic that shows up all over the place in assembly informatics, so I want to make sure everybody knows what a k-mer is. And then we'll move on to annotation. One of the most difficult concepts to understand, but one I think everyone should know about nonetheless, is how we go about finding protein-coding genes in long stretches of DNA sequence. And for that, we really have to talk about hidden Markov models. Hidden Markov models are a really important advance in that area. From there, we'll talk about alignment algorithms, and this is kind of the greatest hits of bioinformatics. People think about sequence alignment as synonymous with bioinformatics, even though that's not the case. We'll talk about substitution matrices. Oh, it is so important to me that people know what substitution matrices are. I often find that people who are pretty advanced in the field of bioinformatics have missed this lecture at some point, and so they don't know what these are. But these are the fundamental scorecards that let us say what amino acid can be replaced by another. It's kind of an important piece to know. And finally, matching to known motifs. We'll talk about the Interpro database that one of our local Capetonians had some hand in developing. All right, so we started our way down this second path yesterday. Today we're moving to the left. We're going to be talking about something that started at shotgun high throughput sequencing, moved through base calling, which operated just the same way we did yesterday with Fred, etc., repeat masking, de novo assembly, gene calling, and functional annotation. Those are kind of the different stops, the functions that we're going to be stepping through today. So we start with assembly. We spent quite a lot of time yesterday on mapping, so making sure everybody can easily distinguish what is mapping for, what is assembly for. That's really valuable because these tend to represent the two forks of how people deal with high throughput sequencing data. So if you've got millions of short reads, that's lovely, but most people are really more interested in the structure of the genome, or at the very least, the structure of a transcript, right? So being able to figure out how these different... Come on in. We have seats at the middle. Or we can seat you on the wing, either way.
Middle.
Middle? Oh, she's going to sit in the middle. All right.
I'll do it.
All right, she's our camera person for the day. All right, so having these reads is fine, but being able to build them into larger sequences through the process of assembly really matters. So we also will frequently use approaches like RNA-seq or something to we'll map those reads to the transcriptome or to the genome, and we find that some fraction of the reads we produce don't actually map to the assembly. You know, it's kind of weird. If you're doing an RNA-seq experiment, you expect everything to code to a human gene, a human protein coding gene, more specifically. But frequently we find that some of the reads we produce from these experiments can't be mapped to the annotation. What if, for example, someone has a bunch of bacteria in them that are producing RNA of their own? Some of these sequencing reads may then come from bacterial strains, not from human. So one of the things that people frequently use assembly for is assembling the stuff that didn't map to try to figure out what coherent genes can be pulled out of that mess. Okay, so we might end up working in a non-model organism. I do work, for example, with hyena, or with chia, or with proteas, or all sorts of things. We don't really have well-annotated genomes for these things. So being able to do de novo assembly, to assemble longer sequences, is very valuable to us because of this non-model organism emphasis. Okay, so as general schematic, I want you to think about two different processes. One is to stitch together overlapping reads. So maybe I have 75 nucleotide from the sequencer here, another 75 nucleotide oligo here, and I recognize, the last 30 nucleotides of this sequence and the first 30 nucleotides of this sequence are identical. So I can use that overlap to stitch those together. So this is the process of forming contigs. Contig just comes from contiguous sequence. So the short reads can be combined to create contigs. But how do you figure out the relationship of one contig to another? If your sequencing reads are all completely independent of each other, you probably don't have a lot of purchase on this question. You can see that this contig and that contig could be set up like this, but what's to prevent them from being like this instead? So this is a big problem when we do single-ended reads. We have an approach that was developed pretty early in the process. You can see I'm actually citing a pretty early paper here, 1991, on this subject. But we have this problem that we have bits of DNA that we've decided to sequence. And in the old days, these were called inserts because they were usually stored in bacterial artificial chromosomes or in cosmids or something like that. So imagine you have this template or insert sequence, and it's got these adapters on either end. The vector sequence just reflects the way you created your library. What you really want to sequence, though, are the two ends of this piece of insert sequence. So most of our sequencing technologies allow us to kind of work our way in from one side. So we can sequence in a little bit from this end, and we can sequence in on the complementary strand from this end. Now we have two patches of sequence that are on the same physical piece of DNA, but they're at opposite ends. Okay? So we haven't got a complete sequence of the whole insert. We have a little bit of sequence on this end and a little bit of sequence on that end, and we simply know these two come from one physical piece of DNA. Typically, the lengths of these DNA pieces can be quite different. You might have one that's 10,000 nucleotides long. And how long a sequence do you typically get from a massively parallel sequencer? Is it 600? 600 represents the number you'd get from an old school sequencer, something like a Sanger. But if you're using something like Illumina, maybe 150, if you've got a fairly long read system. So if you have a piece of DNA that's 10,000 nucleotides long, and you have only 150 nucleotides on this end and 150 nucleotides on the other end, you're in no danger of bridging the whole thing with sequencing. Right? If you're using a PacBio, you could do that. You could get a very long read from that. But if all you have is this piece of DNA that's some length, but you know it's one piece of DNA, and you have a patch of sequence here and a patch of sequence here, you can use that to great benefit in the process of assembly. We're going to talk about how that happens. So this is about how the sequencing reaction is told to get a little bit of sequence from that end and a little bit of sequence from that end. Yesterday in the lab, we watched a video produced by Illumina on how they do sequencing of two different ends of each piece of DNA. So that's an example. So paired end sequencing. Okay, so sometimes the people you meet in grad school turn out to be famous later, and you don't know this at the time. So Jared Roach is one of the most interesting scientists I've ever come across in my career. He's actually an expert Go player. Does anyone play Go? It's an astonishingly complex game to master, and we've only just gotten to a place where a computer can beat a master at Go. This has taken a lot of time. So Jared Roach wrote one of the seminal papers on why paired end sequencing is so much better for doing long assemblies than other tools. So remember that we talked about having a little patch of sequence on either end of a long piece of DNA. So here we're visualizing an individual piece of DNA as a light blue strand, and the regions that we've sequenced of those are shown in dark blue on either end. Okay, so if you project downwards, you can say, all right, I have complete overlaps that stretch from here to here if I stitch together these overlapping sequences. I have completely covered this entire region just by walking from one sequence to another based on these overlaps. But we still have some amount of unclosed gap between these contigs. The magic of paired end sequencing is that because we know that this patch of sequence and that patch of sequence are from the same physical piece of DNA, if this is in one contig and this is in another, we know these contigs border each other. That this one is just a little bit 5' of that one. We may not know the exact length of that piece of DNA, but we know enough to be able to position where these contigs relate to each other. So paired end sequencing is going to help us to understand the relationship of one contig to another. All right, right, now, I find repetitive DNA to be one of the most interesting stories about primate evolution, but we're not really going to go into it at great length. But about what fraction of all genomic DNA for humans relates to protein coding genes? You always give me a number to start with. What's our number to start with today? What percent?
Protein coding genes.
What fraction of the human chromosomes, the nuclear?
Percent. I'm gonna go. There's so much. I'm gonna go. Let's go. I want to say 3, but I think that's too small. But I'm gonna go 3.
He's going to go 3%. Do people want to go higher or lower than 3%?
Isn't there a lot of like?
What wants to say higher?
I'm on the wrong side of this picture.
That 98% is protein coding in the human genome?
Oh, no.
That's what I'm saying. I think it's a little about, but I don't know how little.
Okay, so the number that people throw around loosely is about 1%.
Oh.
Only about 1% of all of our nuclear DNA actually codes for proteins. Now, is that true of a bacterium, say Mycobacterium tuberculosis? No. Their genomes are much more compact. They're very, very small, and almost all of it codes for something, right? So viruses and bacteria have really intense selection pressures that require a great deal less junk in their DNA. Humans, however, are rife with it. And one of the big reasons for it is these repetitive elements. Things like ALU, this primate-specific short interspersed repeat element, inserts itself anew every 20 times someone is born. Right? So we see that we get a new insertion of this sign every 20 births. So this DNA is making more copies of itself as our lives progress. We have bazillions of copies of ALU showing up all over our genome. So there's a real problem with this that we're going to talk about in relation to assembly, and I want to make sure everyone gets it. Because we have these repetitive elements that are taking up some of the space in our genome alongside genes that show up just once, we have this problem that we may make a mistake with where we are in the assembly process. So this diagram comes from a fellow's PhD paper, Batsoglo, who published a paper on the Arachne genome assembler. His PhD thesis turned out to be like the canonical way that a lot of the Human Genome Project was assembled. So he found a way to do de novo assembly on basically desktop computers, and they didn't have Intel i7s at that time. So a very impressive piece of work. So I want you to imagine two hypothetical places in our genome. We have a case where gene A and gene B are close together, but in between them, they have an ALU or some other repetitive element, okay? A and B are close together. Now, way over here, on another chromosome, we have gene C and gene D, and in between them, we have another ALU sequence. Now, imagine that we have sequencing reads here called X and Y. Does everyone see those up there? So we see that the read for X starts in gene A and runs into the ALU sequence. Everyone spots that. So from the perspective of just the read, we see a little bit of gene A and we see a little bit of this repeat. And then over at the right, we see that read Y is from a totally different part of the chromosomal DNA, and it starts in the repeat and continues on through gene D. Now, if you're using this kind of simple bare approach to assembling these reads, you might say, a little bit of read X overlaps with read Y. And you might mistakenly think that gene A is next to gene D. Do you see the problem? Yeah. Okay, so repeats bugger up everything. They're a really big problem. So being able to deal appropriately with repeats is quite necessary. One of the things that's been done is to simply knock out from the sequencing reads anything that is a known repetitive sequence as a way to defeat this process. Okay. So one of the things that's been done is to use sequence alignment algorithms that we're going to talk about in this lecture as a way to recognize all these sites within reads that relate to these repeats. So again, Phil Green and friends at the University of Washington Genome Center created a database called RepBase that had the best examples we had of all these different repetitive elements that appear within human DNA. And then they used cross-match to look for all the sequences that were pretty darn close to these repetitive sequences. And then they could just mark these out and say, okay, these sequences are not to be used as part of this overlap creation process. Okay. Now, that's nice, but we have to imagine that there are efficient ways and non-efficient ways that we can look for overlaps among sequences. You remember that yesterday we talked about inefficient ways that we could map an individual read to the genome, right? You can just Control F on the genome and say, where have I found this particular sequence, and look it up, and then do the next one, and then do the next one several million times. Very inefficient. In the same way, we could say for each read, what other reads in this experiment overlap it by some amount of identical nucleotides. You could do that. But we find that digesting up these reads into k-mers it gives us a lot of power for doing these assemblies on a large scale. So imagine then that we set k to about 25, right? K is just standing in for a number. K doesn't stand for keratin or something here. It stands for some number. So let's set k to 25. That means if we have a bunch of reads that are, let's say, 75 nucleotides long, we would be looking for overlaps of at least 25 nucleotides. So what is the danger in setting k really low? Like, set k to 10. Is there a danger to doing that? Yes, no? People aren't saying it yet. Okay, that's fine. If I have two reads and I'm saying I can overlap these so long as they share even 10 nucleotides, by random chance, I'm occasionally going to match up reads that have nothing to do with each other. Now, if I set it up to 25, the chance that randomly two reads would have these same 25 nucleotides at the end of one and the start of the other is much lower. So we set k to a high enough value that we can find the overlaps. But if you set it too high, let's say if you have reads that are 75 nucleotides long, you set k to 74. Well, that's going to be great so long as you have a read starting at each position in the genome, but it's going to prevent you from finding a lot of overlaps that are legit. So the length of k describes the amount of overlap we require between two reads before we stick them together. So let's now look at what this looks like. Imagine that I have a section of the genome, and I'm showing these 32 nucleotides from it. T, G, G, T, 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 G, A, T, T, blah, blah, blah. We have this region. The sequencing reads in this case are quite short. We're going to just say we're looking at these 25 mers again. So these 25 base pair reads means that I could digest up the reads into 25 length sequences, and I find this one, and this one, and this one. That's all great. So having looked through the reads, the 25 mers that I can pull from it can be overlapped in this way. I want you to think of each k-mer as a point in a graph. Okay, so this TTT, GATT, that is a node in our graph. TT, GATT, that's another point in our graph. TGATT is another node in the graph. Every time we have an all but one overlap for these k-mers, I am going to create an edge. So the edge for TTT to TTG is a directional arrow. I could go from here to here. The node TTG has an edge pointing to TGATT because those two are off by one from each other and share this commonality among them. So you can imagine that the entire sequencing experiment can be diced up into these 25 mers, and we can say, for all of the 25 mers in this entire genome set, which ones are one separate from each other. You can use this to find a way to traverse this gigantic graph of all of these k-mers to put together the longest contigs possible. And you keep going until you've accounted for as many of the k-mers as possible. That is a lot to talk about. Doing it in computer science is... computer scientists often deal with problems like this, and they found some very efficient means for doing so. So the de Bruijn graph, I'm sorry, I do pronounce that one incorrectly as well, is one of these key mechanisms that we use for this purpose. You can see that there are kind of two different techniques that have been competing for this. We used to have things like the Hamiltonian cycle approach for figuring out which k-mers can be linked together to create contigs. But these days, we're using this approach called the Larian Cycle. So this is beyond the scope of this class to understand, but I would have you know that if you encounter something like Larian Cycle and k-mers, we're talking about this process of building very long sequences from collections of really short sequences. Now, some people are really interested in this, and I would really suggest that they look at papers like this one that try to explain how assembly takes place. Modern short-read assembly algorithms construct a de Bruijn graph by representing all k-mer prefixes and suffixes, which is to say that we split up each read into all of the first 25 and last 25 bits of the read, and then drawing edges that represent k-mers having a particular prefix and suffix, those that are joinable in this way. For example, the k-mer edge ATG, here they're setting down to a very small size, just 3, has the prefix AT and the suffix TG. Finding a Larian cycle allows one to reconstruct the genome by forming an alignment in which each successive k-mer, following edge to edge to edge to get each additional letter added, is shifted by 1 position. This generates the same cyclic genome sequence without performing the computationally expensive task of finding Hamiltonian cycles, the old way to do it. So this is a great complexity, and a guy got his PhD for figuring out the process, and this is how our sciences keep moving forward. Our ability to transform huge piles of millions and millions of reads into one contiguous sequence or piles of different contigs was greatly enhanced by our ability to navigate these data structures. Okay, so we also see that Pairedend sequences keep showing up here. Remember that our goal is not just to make contigs, but to understand how they relate to each other. What is their ordering along the chromosomal sequence? So being able, we saw at first our diagram of primers pointing in from either end, and then we saw these light blue lines with patches of sequence at either end. Those were Pairedend sequences. Here they're represented even a third way. We have these little arcs. Each of these represents one physical piece of DNA that has a patch of sequence known from either end. So we're able to learn that, for example, these two contigs are near each other, but they're not as close to each other as these are, or as these are. So we're able to find kind of an ordering that optimizes how these contigs are placed within our assembly. Obviously, we would love to have a complete sequence of everything in each genome. The reality is that that's really only feasible for things like microbes. Once you get to something as complex as even a human genome, we do not have base-for-base resolution for every part of the human genome. And part of that comes from these regions that are really rich in repetitive sequences. Those just bedevil assembly and have created a lot of problems. So our assembly of the human genome gets better all the time, but it will probably never be completely perfect. Okay, this gets us to the intermission. However, we have 0 time, but I stayed within 30 minutes for that first part. I'm very, very proud of that. Okay, so we talked about k-mers, we talked about assembly. All of that was a process of getting from a bunch of reads to contiguous sequence. But, and this is a really important consideration, a genome is not just a long sequence. If you don't have landmarks placed along the sequence, you don't have a completed annotated genome. By the way, I realize I didn't let you know because you came in right at the hour. We took quizzes at the start. We'll have you take the quiz at the end. Is that okay? Yeah. Okay, I didn't want you worrying about that. Okay, cool. All right, great. On we go. Now, hidden Markov models. Given a strand of DNA sequence that's several thousand or millions of letters long, how do you figure out the parts you really care about? As we said at the start of the lecture, we don't really, not all parts of the genome are equally important to us. Generally speaking, someone like me is going to care a whole lot about how many protein coding genes there are, rather than where are all the telomeres? And yet there are people in the department who care a lot about telomeres, right? So each of us is going to have a different kind of thing that we want to find within the sequence. So one of the most powerful methods that we have to use is kind of a machine learning technique, really. And it's the hidden Markov model is a way to represent a complex biological feature based on different elements that it possesses. I'm going to go ahead and pull this shut if you don't mind. Jackhammer doesn't have to invade every space we have. So I want you to think about the complex structure of a protein coding gene. Right? We have things like a promoter, we have an enhancer upstream of that, we have introns and exons, we have a start codon, we have all kinds of different features. These biological features can be represented by different data structures within a single hidden Markov model that understands how these pieces connect. So let's start with kind of a more abstract take. A hidden Markov model is represented in this case by these red items. What we have is a directed graph. In this case, we're going to start at state 0 in that red thing. We're going to work our way down through either state 1A or state 1B. Those both feed into state 2. State 2 can either come back to itself or it can lead to state 3A or state 3B. And state 3A and state 3B both end up in state 4, at which we've hit the end. All right? So that's a very abstract way to think about it, but this is important, I think, for us to get what's going on with hidden Markov models. As we progress through the different features of this model, we have transition probabilities that state whether we're going from state 0 to state 1A or from state 0 to state 1B. Does everyone see those edges coming down from it? The one on the left has a transition probability of 0.8. The one on the right has a transition probability of 0.2, which means that 4 out of 5 times that this feature is encountered, we go to state 1A rather than state 1B. So that's a transition probability. But it's called a hidden Markov model because we don't actually get to see which part of this model we're traversing. What we have is a visible sequence that we think of as being generated by this model. So to use an example, if you have a promoter for a gene, what is the sequence most likely to correspond to a promoter? We have a nickname for promoters, at least in the microbiological world. Has anyone heard the term Tata box? Tata box. Okay, good. It's a silly name, isn't it? All right, so a Tata box is characterized by the sequences that are associated with the Tata box. And the Tata box is comprised of what 2 letters most dominantly? T and A. Okay, very good. Right. So the T and A letters that appear in the sequence, in the DNA sequence, it's not RNA, right? Because it's T, right? Not U. So we have a region of the sequence that is rich in T and A nucleotides that we say, that's a Tata box that is part of the promoter for this gene. So what I would like you to think then, in terms of the hidden Markov model, is that if you have a box in your graph that represents a promoter, it's quite likely to output or emit, as we say in this terminology, T or A letters in the T or A nucleotides in the sequence. Okay, so we've talked about transition probabilities, which refers to how we move from one abstract part of this model to the next. But we also have what appears in the sequence that is characteristic of this part of the model. So if you have a promoter, its emission probabilities are really rich in A and T nucleotides, and quite a lot lower probability for C and G nucleotides. Okay, so that's emission. So we can imagine then that this biological feature may, in one instance, produce sequence 1, where letter 1 is emitted by state 0, letter 2 is emitted by state 1A, letter 3 is emitted by state 2, letter 4 is emitted by state 3B, and finally letter 5 is emitted by state 4. But you remember that in one case, we had a quick cycle within this structure. See how state 2 has 3 different possible next states? State 2 could be followed by what in this red box graph? Just look at the arrows. What are the red boxes that it leads to? Right, so state 2 could give rise to state 3A, or it could give rise to state 3B. But we see that that only happens what fraction of the time? Half, yes, exactly, right. So yes, state 2 to state 3A happens 1 in 10 times. State 2 to state 3B happens 40% of the time. But what accounts for the other half of the time? Itself, exactly. This is a cycle. So if you were in state 2, you may continue on in state 2 for the next letter that's emitted. So we see that example sequence 2 is also possible. We see that state 2 could emit letter 3, cycle back to itself, and then emit letter 4. So we have to be cognizant of all of these different transitions that are possible. Okay, so that's fairly abstract. Let's now talk a little bit about the algorithms that relate to this. So Baum-Welch is the strategy that we use for training a hidden Markov model. We have a whole bunch of sequences. Those sequences have been marked up to say this is the part that relates to state 1, this is the part that relates to state 2, and so on. We've dissected these sequences out and said, this is the structure that we interpret the sequence to represent. So Baum-Welch is going to train a hidden Markov model to learn all of these emission probabilities and transition probabilities. Now the forward-backward algorithm is a test of our HMM. We have our HMM all nice and trained now from Baum-Welch, but we're going to apply the forward-backward algorithm because we're giving it a new sequence, and we're going to ask, what's your interpretation of this? How would you, is this sequence one of these things that we've shown you as part of the training set, or is it not? So that's going to give us the best probability that this sequence is a match to this model. And finally, Viterbi allows us to say, not just, is the sequence one of these things that this model represents, but how does it diagram out which parts are further promoter, which parts are the intron-exon boundaries, and so on. So having trained our HMM with Baum-Welch, we use the forward-backward algorithm to say, does this new sequence represent one of these? And finally, we can use Viterbi to say, how does this structure map onto the sequence we've shown? I am not going to try to explain how these work. I personally find it very bewildering. Okay, so we know what protein coding genes are like, right? This is a computer scientist's view of what genes look like, protein coding genes. And I note I'm only showing you half the model. The other half of this model we usually show right below this, and it reflects if the gene appears on the complementary strand. So we have to consider that the promoter is at this end, and then we have our start codons, and then we have our exons separated by introns, and so on. But we could also be screening for genes on the other strand, and for them, the final exon appears before the promoter, because we're seeing it all in reverse order. Okay. So we can imagine then that we're sitting in intergenic region. We spend most of our time there because most of the sequences in the genome do not correspond to protein coding genes. Eventually, we encounter something that twigs this part of the model marked promoter. So maybe this is a whole bunch of T and A nucleotides. We get to the 5' UTR. Does anyone remember what UTR is? Untranslated region, right. So this is a part of the gene that doesn't actually make it to the ribosome. Well, sorry, that would get to the ribosome, I guess, but it wouldn't be actually translated, of course. Some of the genes that we have just one exon in them. They have a very simple structure. So for something like that, you're simply going to go from intergenic region to promoter, and translated region to a single exon out to the 3' untranslated region. You've got a poly A signal, and then you're right back in the intergenic region. That's a really simple course. But sometimes we instead find that we have multiple exons and introns. And in this case, the software has to get really pesky about which explicit kind of intron-exon boundary is concerned. Because you may have an exon that starts right at a codon boundary. You might have an intron-exon boundary that comes one nucleotide into a codon. Or you might have an intron-exon boundary that comes 2 nucleotides into the codon. So the software tries to keep each of those straight. That's intron frame 0, intron frame 1, and intron frame 2. So if you have a multi-exon gene, you spend some time ricocheting around in the top part of this hidden Markov model, before you hit your final exon, do your 3' untranslated region, your poly-A signal, and come back to the intron, to the intergenic region rather. So that's a complex data structure, but it reflects this knowledge that we have of what genes are like. We frequently find that genes have multiple exons, and we need to be able to recognize not just that this megabase contains some gene, but where the promoter lies, where are intron-exon boundaries, and so on. It's a difficult proposition. Hidden Markov models are our best guess for how to model this appropriately. And I would note that they're pretty aggressive, frankly. When you run this on genomic sequence, you quite frequently get back hits on things that are not considered legitimate protein coding genes. So you have some number of false positives that result from this, but this remains our best strategy for picking out where these protein coding genes are within the genome. I see a question? Oh, just to shake you, okay. Great. This brings us to sequence alignment, which is definitely one of the greatest hits of bioinformatics research. Why do we need to bother with alignment of sequences? We start with orthologs. Understanding the difference between orthologs and paralogs, I just warn you, is something I often write quiz questions about because it's a really important piece. If you have the same gene showing up in multiple species, it didn't get there by chance, right? A long time ago, we had a common ancestor with yeast, for example. As a result, if I look at a gene for a gene that's part of the glycolysis pathway. The glycolysis pathway has been in use by living organisms since we and yeast had a common ancestor. And so the gene that we use for chopping apart glucose is very similar in sequence to the gene used by yeast to break apart glucose. So that means that if I look at the protein sequence of the yeast enzyme and my enzyme, I can still see sequence similarity over the span of billions of years. It's kind of astonishing, but it's true. You can do that. If I were to compare my genes to that of a chimpanzee, I'm going to see that the relationships are much, much stronger. They've had much less time in evolution to separate one from the other. In fact, you don't have to just deal with, instead of simply relating to protein sequence comparisons, I could even look at the DNA sequence itself for those genes between me and a chimpanzee in the same glucose-breaking enzyme. So that's brilliant. So recognizing orthologs is very, very valuable because one of the first things you're going to want to know when you've sequenced a new non-model organism is how do its genes differ from its near neighbors. And so you may know that these 100,000 features have been called putative genes by a hidden Markov model, but that doesn't tell you anything about their function. You're going to learn about its function by blasting it against nearby species. Okay, so orthology is a really valuable thing for us to learn. Virology is also very important. Over the course of time, many genes in our genomes, and certainly in tree genomes, they're just kind of ludicrous on this score, will make many copies of particular genes. And over time, some of those gene copies will go away, while others will take on new functions within the developing organism. So one of the if I remember correctly, one of the crystalline genes that we use in our eyes is actually a gene duplicate from an enzyme that operates in the cytosol. So over time, this gene copy has drifted way apart and has taken on a completely new function that was not necessary to the organism at the time that the gene duplication event took place. So we call those paralogs. Within our own genomes, we have multiple copies of some genes that have drifted apart from each other in function, typically. And conserved regions. Generally speaking, biologists spend more time focusing on parts of the genome that are under selection pressure to stay exactly the same than they do looking at things that are just changing randomly. The suspicion here is that parts of our genome that don't change over evolutionary time are playing some sort of function, whether they're part of protein coding genes or not. Okay, so looking for parts of the genome that don't change is our tip-off that they are important. Okay, so there are lots of different terms that get tossed around on sequence alignment. I'm going to try to walk you through the key ones here. global and local alignment. If you have a complete protein coding sequence and you want to compare it to another complete coding sequence, let's say a cat enzyme and a hyena enzyme, you have these protein sequences, you are probably trying to do a global alignment to say, how does the sequence we know for cat compare to the one that we've observed in hyena? If, however, you are looking through protein, a very long stretch of genomic sequence to try to find features that are conserved from one genome to the next, you're probably doing something more like local alignment, looking for little subsections of this long stretch of DNA that map to short subsections of this other organism's piece of DNA. So that's a global versus local distinction. Now, optimal versus heuristic is also kind of a difficult concept, but I would say that sometimes we have algorithms that are built around the idea that we want to get the very best scoring alignment possible between two sequences. Something that is provably the best alignment there. Something like Smith-Waterman is one of these sequence alignment approaches that matches that. So we tend to call those algorithms, provably correct algorithms and so on, but often those algorithms are a lot slower than those that would give us what is likely to be the best match. So I think most people in this room have probably heard of BLAST at this point. Okay, BLAST is definitely one of the big hitters in the bioinformatics space. BLAST does not provide you the provably best match in the database. It does, however, produce what is likely to be the best match in the database. And that difference between the provably best match and what is likely to be the best match allows BLAST to be much faster than if it were giving you the provably best match. We'll talk about how that works in a minute. Evolutionary distance. If you have organisms that are very closely related, you should probably be doing your comparison on the basis of nucleotides. If, on the other hand, you have a very distant evolutionary relationship, remember I was talking about yeast and humans? I mean, our common ancestor was billions of years back. So trying to do a comparison on the basis of nucleotides is really problematic. It's much better for us to do those comparisons on the basis of amino acid sequences for proteins. Okay, so the substitution matrix, one of these concepts that just keeps on giving, is the scorecard that we use to evaluate whether these relationships matter or not. So odds ratios are something that show up all the time in our field. And generally, these relate to a particular comparison of two probabilities. So how many people in South Africa died of lightning strikes last year? Okay, I'm going to look to this back corner, the people sitting near the power tap. Would somebody like to make an estimate of how many people died of lightning strikes in South Africa last year? Just give us a number. It doesn't have to be perfect.
I mean, you're not going to be wrong.
15.
I mean, you are
15?
wrong, but
15 is a good number, I think. We'll go with 15? People okay with 15? Think it's higher or lower?
Higher.
Higher? How much higher? You want to go to 50?
Maybe 9.
What's that?
Maybe 9 or 10.
9 or 10? Okay, well, I think that number's fine. Would we accept that it's less than 100?
Yes.
Okay, great. All right, so just for ease of computation, I'm going to say 55. The number doesn't really matter. It's a small number, relatively speaking. How many people in South Africa? About 55 million? Okay, so if we have 55 million people in South Africa, and let us say 55 died of lightning strikes, that means the chances of an individual in South Africa dying of a lightning strike is?
why didn't you get 1 million? Is that right?
Wait, no, I'm sorry. We said 55 people died of lightning strikes. We said that there are 55 million people in South Africa. 1 in a million per year. Okay, 1 in a million per year. Great. So that is a probability that every person walking around here, no favorites played here, has a 1 in a million chance of dying of a lightning strike in South Africa next year. Okay. Now I have to say I'm terribly biased against these shuttle bus taxis. I think those guys are really bloody dangerous. So let us now try to frame an estimate. How many people died in shuttle bus taxi accidents in South Africa last year? A lot. Okay, it's a lot. But how much is a lot? You want to give? Are we at 1,000? 10,000? I'm sorry, I picked on them for the first part. Anyone on this side of the room want to give us an estimate of how many people died in shuttle bus taxis last year? It doesn't have to be accurate.
3,000.
What's that?
3,000.
3,000. Whew. 3,000 people. You know, a shuttle bus taxi driver was shot by one of his passengers at my intersection last year. So I'm just really sensitive to this issue. All right, so 3,000. Okay. Does anyone have some estimate of what 3,000 divided by 55 million is? Doesn't have to be accurate, it just needs to be a number. Shall we say it's 1 in 10,000 people? Is that an okay guess? All right, let's return to the topic at hand. Dying of a lightning strike is likely to be a 1 in a million event. Dying in a taxi event, on the other hand, is more like a 1 in 100,000, 1 in 10,000 event. So we can use an odds ratio to compare these two, but these numbers are both really small. If it's a 1 in 10,000 event versus a 1 in a million event, how much more likely is one than the other? 1 in a million versus 1 in 10,000. 100, very good, yeah. So we can compare these two numbers and say 1 is 100 times more likely. So the next time you see lightning gathering in the sky, well, worry about it and get under cover. But the chances that you personally are going to die from a lightning strike is much lower than one of those minibus taxis winging you as you try to walk across the street. They just terrify me. Okay.
I saw the funniest thing this morning.
Oh, no time. But we can do really funny things afterwards. We will do afterwards. Okay, I'm sure it's going to be a really, really great story. We are sitting at 8 minutes. 8 minutes is all I've given myself here. I'm so sorry. Okay, so these numbers can be expressed as a probability divided by a probability. That's an odds ratio. But we would frequently find that our numbers get a whole lot more sensible if we put them on a log scale. So 1 in a million is 10e minus 6. 1 in a million translates to e minus 6. The 1 in 10,000, I think we said, was e minus 4. 1e minus 4. So on a log base 10 scale, these are 2 in difference. Because 2 to the 100 to the second power, 10 to the second power is the ratio between these two. So we frequently find that comparing these low likelihood probabilities gets a lot more sensible when we put them on a log scale. This slide is about that math. What does it look like? This is one of the most commonly used scorecards in all of bioinformatics. I always feel that people need to know more about this than they really do. So fair warning, it's just like quiz territory for me. So this is the Blossom 62 comparison to ask which amino acid could get replaced by another in evolutionary terms. When we compare a bunch of sequences that are closely related to each other, which amino acids typically replace others? If you see a positive score, that means this replacement happens more than you would expect by random chance. If you see a 0 here, it means this replacement happens just about as much as you would expect by random chance alone. If you see a negative score here, it is telling you that we observe this substitution much less frequently than we would expect by random chance alone. So when we look across here, we see that we've got a high value over here at, I believe, 11. What is W in amino acids? Tryptophan, exactly, right. So tryptophan stays tryptophan much, much more frequently than we would expect by random chance alone. I can just go from tryptophan on this row and tryptophan on this column, and they align right here. This is a log base 2 scale, which is saying that tryptophan remains tryptophan 2 to the 11th fold more than you would expect by random chance alone. Tryptophan is obviously not that replaceable. Okay. But let's look at substituting one letter for a different letter. So I've highlighted in yellow all of the values that are greater than 0 that are not on the diagonal. What's the highest number you see there? I see a 3. Okay, so what is this 3 representing? It means that we're replacing isoleucine with valine, or the other way around, valine replaced with isoleucine. This table can be flipped on its axis and it's identical. So this is to say that isoleucine and valine tend to be very much replacements of each other. Consequently, if you saw an amino acid change in your protein in one strain to another from valine to isoleucine, it's probably not making that big a difference. What about serine? What do you see for serine? If you look down the serine column, you can see that serine can be replaced by threonine. It happens about twice, 2 to the first power is 1, right? So it happens about twice as frequently as you would expect by random chance. We see that serine can be replaced by asparagine, which is a little more surprising, or serine could be replaced by alanine. Now, I would note that if serine becomes alanine, it's no longer a phosphorylation site for the protein, so that's actually a very big change. But not all serines are places that get replaced like this. What about methionine? Methionine and leucine are pretty good comparisons for each other. Methionine and isoleucine, also allowable. But really, methionine and tyrosine are pretty replaceable. So how does this work? How do we use this scorecard? I'm going to move on to the next slide here. What I'm showing you is an alignment of 2 sequences. We've got the Regatta sequence and we've got the YDG sequence. And I'm asking the software, I'm asking Blossom, how do these score against each other? So for R and Y, I'm going to look for the Y column. Here's the Y column. And I'm going to look for arginine. Where's arginine? Way up at the top. Okay, so arginine and tyrosine have a -2. Does everyone see that in the table? So I just write a -2 between R and Y. Next, E and D. What do you guys feel about that one? Is aspartic and glutamic acid, are aspartic and glutamic acids good replacements for each other? They're both acidic residues, right? So that's something you would think would score pretty well, and it does in fact score up +2. So for each of these pairs of amino acids, I'm going to look up the score, and then I simply add them all together. That's how Blossom 62 works in scoring a particular alignment. The sum of across there gives us some way to evaluate whether this is a good match or a poor match. All right, now, if you're using something like Needleman-Wunsch or Smith-Waterman, they're going to use this dynamic programming technique to figure out whether you want to put in a gap here or simply align these along a diagonal. Anytime you see something like this, this diagonal here, it's saying just use these one-to-one versus each other. When you see a jump horizontally or vertically, like at this little bootleg here, it's saying it has to insert a character in one of those sequences to force the best alignment possible. So this is a great approach to doing this kind of alignment, but it is slow because it's producing a provably correct result. The other thing is that Blossom 62 doesn't say anything about how we are to score gaps. Now, gaps are a pretty big deal because they require an insertion or deletion in order to produce them in the sequence. So we have to have some way that we score those gaps alongside Blossom 62. But BLAST has become this approach that just dwarfs everything else. There are lots of algorithms out there since BLAST was published that are faster or produce slightly more accurate results, but BLAST is definitely one of the big gorillas in this space. These papers have wrapped up just a really tremendous number of citations. I think it's in the hundreds of thousands at this point. At this point, you just use BLAST in your paper and you assume everybody knows what you're talking about, but it's actually kinder to bioinformaticists everywhere to cite the papers that introduce the tools you use. Just saying. Okay, so BLAST is fast because it assumes similar sequences are going to contain some regions of identity. Similar is not the same thing as identical, right? We all get that. So if you were simply to score sequences based, giving them 1 point for every identical amino acid and no points for every place that was other than identical, that would be that is a homology score, but it's really an identity score. It's only giving you a match if they're identical amino acids. So BLAST is starting with the presumption then that any biologically related sequence will have some amount of identity matching between them. And it calls these seed matches. It's going to require in protein sequences, for example, that 3 successive amino acids will be identical between the two. Actually, in later versions of the software, they require that all protein matches have at least 2 regions where 3 amino acids in a row are identical between the two. So those are seed matches. These seed matches are the starting point for the software. It's not scoring on how many seed matches there are between pairs of sequences, but rather on something called a maximal segment pair. This is to say, some part of the sequence that has a very high degree of homology, building upon these regions of identity. So I'm going to show this, sorry, there's one other concept I really need to cover on this slide. Expectation values are a really, really important characterization that we use in a lot of bioinformatics, so I'd like everybody to know this. One of the principal scores that comes back from the use of BLAST is the expectation value. This represents the number of alignments with scores at least this good that should have been expected to have occurred by random chance alone. All right, so if you get back an expectation value of 1 from your BLAST search, it means the software says, gosh, well, I would have expected that your best hit, if there were no related sequences, would be about this good. So a 1 is a terrible expectation score. You should not trust any hit of 1 on an expectation score. It means this is about what you should have expected to have occurred at random. All right? So expectation values are very, very frequently used as our way to score how good a result came back. I love this slide. It's always a lot of fun to navigate. But this is a finite state machine. It represents a way that we can compare a sequence to a whole database of sequences to say which sequences in this database contain any 3 consecutive amino acids that are identical. It's actually a beautiful little bit of code, and I'd love to walk you guys through it, but I don't have time. Okay, so graphically, when you give BLAST a sequence and a sequence database, it has to do these comparisons between individual sequences kind of one at a time. It's only going to consider that subset of database sequences that contain identical seeds between them. So here I've got RPE that I've marked as a big wide red block. That's a seed match because RPE is found both in our query and in this database from the sequence from the database, sorry. We also have MCT down here. So MCT is identical between the sequence we gave at the query and this particular entry from the database. But the maximal segment pair is bigger than that. You can see, for example, that phenylalanine is conserved between these 2 sequences. It's identical. We see that asparagine is identical between these sequences. So those are obviously really nice to include in our maximal segment pair if we can, but we're going to have a little bit of cost to do so. Now, if we look at these regions between MCT and RPE, we see that VDV in the lower sequence has no identical amino acids with AEA. But remember, we said valine and alanine are frequently found to replace each other in the sequence database. So the score does rise for the inclusion of these AV pairings. E and D, we also observed in the BLOSSOM matrix, are quite frequently replacing each other in biological sequences. So that's going to get more positive points. As we move to the left here, E and V, they're not really good. I mean, you don't get a positive in BLOSSOM 62 for combining those two. F versus L, yeah, okay. It's fine. It adds to the positive score. As we move to this side, serine and alanine, we saw those as well. That's a positive. K versus H, well, they're both basic residues, even though their structures are quite different. So that's a plus. E versus D, great. But when you get to something like proline versus glutamic acid, they're really different biochemically. That's going to give us a pretty negative score. So the process of forming this maximal segment pair, the MSP, means that we need to extend upon regions of identity, of perfect replication between the sequences, to include things that are homology, are net positive on homology, but not so much so that we end up running through a whole bunch of things like proline versus glutamic acid. So we start with seed matches, we build out from those a maximal segment pair, which is the largest region of high score that we can get for this particular pairing. That score for the maximal segment pair then represents this particular sequence match in the computation of the expectation value. Okay. Multiple sequence alignment is a very good thing. There are lots of tools out there for it. I'm not going to discuss them to a great extent. Structural biology, again, is not something I'm going to talk about a whole lot, but I would simply note that the reason why we are so want to find regions of homology among different sequences is our belief that they're functionally related. And that's troublesome because we have this little step to go through. Just because things have the same sequence, we usually infer that they take on similar folds in protein structures. I think you've talked with Colin Kenyon a little bit on this subject. Okay, he's a genius in the space of structural biology, so he's a great person to ask about this. But what I want everyone to internalize here is the central dogma of structural biology, not the molecular biology side. Things that have similar sequences are likely to adopt similar three-dimensional folds. Things that have similar three-dimensional folds are likely to take on similar functions. So if we find that proteins in two different species have similar sequences, we infer that they fold similarly. If they fold similarly, we infer that they function similarly. Okay, so finding regions that are known to be conserved across lots of proteins gives us some notion that these different proteins are functioning similarly. PSSMs, position-specific scoring matrices, are one of the ways that we go out to find these comparisons. Oh my gosh, it's already past the hour. I'm glad no one has chased us out just yet. I'm going to just keep going, so I'll try to keep it compact. We now have something called a leucine zipper. A leucine zipper. Leucine zippers create a whole bunch of adjacent basic amino acids, and one of the things that this enables them to do is to interact with DNA, which we should all remember is in fact an acid. So these basic residues are able to interact with the acidic structure of the DNA in order to carry out its function. If we look at many, many examples of leucine zippers in the protein sequences we've got, we find that this particular motif, sequence motif, is very frequently found. And we believe that these basic residues, lysine, arginine, I think there may be a histidine here somewhere, but generally these R's and K's are part of this basic region within the protein that interacts with DNA. So we're able to stack all of these sequences on top of each other through alignment, because alignment is finding this region of homology. And we can see that some of these have very high conservation for a particular amino acid. For example, right here, we see that arginine, all up and down this column, is completely uniform, which, remember I said conserved sequences are kind of a dead giveaway, that this part is strongly evolutionarily conserved. And that's probably because it plays some essential role in the function of this motif. Okay, so you can make some sort of statistical statement about different positions within this alignment. For example, this position is not all arginine or all lysine, but we can say from looking across many different structures that lysine shows up in 1, 2, 3, 4, 5 of the cases, and arginine shows up in 1, 2, 3, 4, 5 of the cases. So we could say this position has a 50% chance of being K and a 50% chance of being arginine. The value for that is that once you've established a signature for what this looks like, this is a sequence motif that matches this structure, you can express it as a POSSUM, a position-specific scoring matrix, so that you can score new proteins that come down the pipe to evaluate whether they contain this structure or not. So from You'll very frequently see in bioinformatics that we are framing a search where we want to find more things like this. This teaches the presence of this sequence motif in all of these sequences gives us a signature, and we can then use it in new sequences to say, does this or does this not contain a leucine zipper? One of the most powerful resources we have for making this kind of search is called Interpro. So if you are trying to understand a particular gene that you've been asked to clone for class or for your projects, something like Interpro is going to be very valuable to you. Because Interpro is not simply going to say, what protein is this most like, which is what BLAST will do. Instead, it's going to dissect that into particular motifs and domains that fall within that sequence. And that's very valuable when you're trying to understand how this protein does its job. So this is an example of a protein called castor bean ricin. Has anyone heard of this? Okay, one of the last things I did when I was back in the United States was to play part in prosecutions by the Department of Homeland Security. The Department of Homeland Security has a very strong feeling about ricin. If you are producing ricin, they believe you are up to no good whatsoever, because you can kill people with ricin. There's actually cases where spies have killed people in other countries by putting a needle on an umbrella tip and lacing it with ricin and just giving them a good poke at the subway station. It's that dangerous, right? But ricin is not an exotic protein. It comes from the seed coats of castor beans. And people use these they plant these as shrubbery in landscape all the time. So, odd fact, in the United States, if you grind up castor bean seeds and run it through a paper towel, you are a criminal. Because you have committed a biochemical process by filtering it through a paper towel en route to producing ricin. Don't do that. Okay. Anyway, I'll tell you about the experience of serving in a trial sometime. It's fun. Bought my car. All right, so here we have a protein that has various domains within it. We have different links to structures made available to us. The Gene3D database has that. But we see that we have these different ribosome inactivating proteins. These are domains within the protein sequence. The really fun thing is that the ribosome inactivating protein sequence has the abbreviation RIP, because what happens to something if you turn off its ribosomes? It's dead. It's way dead, right? So the ribosome inactivating protein prevents that organism from producing any more proteins. But how does it get in, right? So ricin is this amazing protein. What's going on at the C-terminus? What do we have at the C-terminus of this protein? Anyone see it? Okay, C-terminus is the far side. It's going to be one of these SSF and PS5 SMs and so on. That is a lectin domain. Does anyone recognize the term lectin? What? They've heard it. Okay, someone's heard the term lectin? Okay, what I would have you know is that a lectin is a sugar binding domain of a protein. So what we have is one protein that combines the ability to bind to a sugar and a ribosome inactivating process. Cells frequently label themselves with sugars as a way to enable them to interact with other cells. So when ricin appears in someone's bloodstream, it looks for cells that have these sugars on the surface, bind with the sugar, get endocytosed by the cells, and then it kills all the ribosomes. So in one protein, we have the functionality to get into cells and the functionality to kill them. It's kind of a shocking little protein. So you can search for any uniprot protein in Interpro, and it'll give you the pre-baked set of domains that were found when you did it. And of course, you can give it new protein sequences and get its assessment of what they represent. A very powerful service. Nikki Mulder over at University of Cape Town, who runs the computational biology group, was one of the developers behind this amazing framework. Okay, lots of takeaway messages. I realize that was a really, really fast tour of assembly and annotation, but I hope you've got a whole bunch of great key concepts to take away. Assembling genomes is greatly aided by the use of Pairedend sequencing. Those Pairedend sequences help us to understand how these contigs relate to each other in the sequence order. repetitive sequences can make distant features mistakenly be mistaken for being very close together. So we want to make sure that we recognize these highly redundant sequences before we go about doing our assembly process. Hidden Markov models, they are complex, but they are one of the dominant ways that we discover which areas of a genomic sequence are protein coding. So they really do matter. Aligning sequences was the original killer app of bioinformatics. The ability to do something with these sequence databases that were being accumulated by major repositories was hugely important. And having tools like FASTA, tools like BLAST, was really essential for our being able to compare new sequences to those that were already in the database. However, those are heuristics. We're not guaranteed to get the best scoring sequence in response. It just seems it's generally quite likely that we do so. Okay, that is this morning's lecture. I will see you again this afternoon at 2 o'clock, right back in this room again. So thank you very much.